speak it as we go and describe information as we go, so you can start drawing the pieces to it as I'm going through it, and that will help you. All right, so most of you guys know what white blood cells are for, right? They're infection, immune system of our body. So that is part of the lymphatic system. That is part of the lymphatic system. Okay? So the lymphatic system consists of what? Our lymph nodes, right? And lymph nodes are typically where we'll find things like these lymphocytes, okay? Like lymphocytes, and this kidney bean-shaped nucleus would be representative of what? Big, large nucleus. Macrophage, right? Exactly. A macrophage, or monocyte, right? So this is part of this, right? So where do we have a monocyte? So this says macrophage, but that's what it comes. So monocytes are in our blood, and then when they leave the blood and come into the tissue, then they're called macrophages. Okay. So, but the mnemonic here is what?
Never let my engine blow 60-30-830.
Okay, so never let my engine blow 60-30-830, and this is the percentage of white blood cells in your blood. Okay, so you guys that are going into nursing and all this stuff, this will come back, and you'll see this often. Okay, this is when you do CBCs and CMPs. When you start looking at your blood work, you can start determining what type of infections you have by the increase in the numbers. Now, just going by this information right here, when we have an infection, what's the first white blood cell that shows up to the scene?
Neutrophils.
And why would a neutrophil show up first?
Because they're the most abundant.
Most abundant. They're also the most motile. In other words, they move the best. Okay. So when we start having an infection, so we have an infection that comes in your mouth, attaches to the inside lining or the mucosal lining of your, say, your throat. It starts to damage the tissue as it goes in. All right. As that happens, the tissue starts to swell. Fluid, that's fluid, right? Swelling is fluid. And then once that occurs, then these neutrophils and monocytes will actually leave the blood through a process called, write it down, diapedesis. Diapedesis. Or immigration, not with an E, not an I. Immigration.
How do you spell the other one you said?
With the pen. D-I-A-P-E-D-I-S-I-S. Diapedesis. I'll write it down. So, diapedesis. Okay. All right, so that's the process of where those things will actually leave the blood and come into the interstitial fluid. Remember, that's the fluid around the cells. Okay. All right. So the first thing that shows up is what we call a neutrophil. Now, we've already said that a monocyte that comes into the extracellular fluid is called a macrophage. So if we have a macrophage, then we probably have a microphage. And a microphage is a neutrophil. Okay? So a microphage is a neutrophil, a little eater. Okay? Now, a neutrophil is a non-presenting cell, and I'll describe that in a minute, and a macrophage is a presenting cell. Okay? That's a process that's this process that we're going to talk about here. Okay? All right. So of course a neutrophil is representative by what? How many lobes does a neutrophil have in the nucleus?
4.
2 to 6. Right? 2 to 6. So when you show a cell that has multi-lobe nucleus, typically you're describing a neutrophil. Okay? All right. So what I'm showing you here is this process of bringing a bacterium into the cell is what? What is it called when we eat something?
Go ahead. Phagocytosis.
Phagocytosis. Okay? So when we eat something, it's called phagocytosis. Okay? All right, now, not when the cells eat something, it's called phagocytosis. All right, so now this, remember we have lysosomes, peroxisomes, all these somes, which are vesicles, right? When we bring in this bacterium, and I've showed a bacillus here with a tail, right? When we bring in this bacterium, that vesicle with the bacteria that is eaten is called a phagosome. Okay, so go through the process is phagocytosis. The thing that you brought in is called a phagosome. Okay? Remember, that's just a pouch of something that you've eaten and brought in. It's a phagosome. Okay? All right, so now what happens is this neutrophil, and I'm showing you this green little vesicle, which is a pouch, right? And this pouch happens to be filled with digestive enzymes. So it's a, what's the pouch in the, what? Nope. The pouch with digestive enzymes in it is called a, what? Lysosome. Okay? All right, it's called a lysosome. And that lysosome will then release those digestive enzymes into the phagosome, which will then destroy the bacterium and leave nothing but what's on the outside of every cell to determine whether it's self or non-self. That? Say it again. Antigens. Okay, antigens. So you have these markers on the outside of your everything, and they will determine whether it's self or non-self. If it's non-self, your immune system should attack it. Okay, if a normal immune system. Okay, if it's yours, hopefully your immune system is not attacking it. Right, that's what we call autoimmune diseases. Right, like rheumatoid arthritis is an autoimmune disease where your body starts attacking its own joints. Okay, all right, that is an immune system problem. Okay. All right, so we have the bacterium will be destroyed, and then through a process of, how do we release stuff out of the cell? Exocytosis. I know I'm bringing up all these old things. It's just killing me, right? Anatomy 1. So we release these antigens. Remember this, and I'm just showing you a star antigen. Of course, there's no such thing as a star antigen. It's just representative of a protein. Okay, all right. It makes it easier to describe it. Okay. So also, these bacterium are multi-antigenic. Okay, so they have a whole bunch of different antigens on the outside of their cell. So it's not just one that gets released; it's a whole bunch. And our immune system works on a whole bunch of different antigens all at the same time. So it's never about one and one. But I got to show you it in one. It makes it a lot less complicated. Okay. All right. So just remember, we have multiple antigens on the outside. Okay. All right, so those antigens are released into where? Okay, so where is this occurred? Where did this neutrophil come in, and where did that occur at? The site of infection. Okay, so common sense. Where did it happen? So if you have a sore throat, and this is where your infection is starting, it's in the interstitial fluid of your mucosa of the throat, okay? So that epithelium, maybe a little deeper than that, hopefully not too much, right? Because then we start having systemic problems. But anyway, so we have this occurs there, and then it releases those antigens into what? I want you to make the connection here. That's why I'm pausing here for you, okay? It releases it into what? Where's that cell at? In the what?
Where the inflammation is, the interstitial fluid.
In the interstitial fluid, exactly right. Okay, so it's not into the circulatory system. It doesn't release them into the blood. It releases it into the tissue. And if it's in the tissue, where does it go from there? What? Okay, it's already at the infection. Where's it going? So where does interstitial fluid get picked up and taken back to? No. So what is interstitial fluid? What happens when you stand too long? Your feet swell up, right? Where does that fluid go? How does it get from your feet back to your circulatory system? Say the lymphatic system. Okay, so the lymphatic system. So it goes from your interstitial fluid back into the lymph vessels, then where to? Then to the lymph nodes. Okay, so the lymph nodes. So I'm trying to make you put this together. so that you can, this is trying to help you with the practical too. All right, so anyway, so these antigens then will go from the fluid, the interstitial fluid, into the lymphatic system, from the lymphatic system into the lymph node, where we have a conglomeration, a huge buildup of, and this is a lymphocyte, although the nucleus is much smaller than normal, because normally the lymphocyte is huge, right? The nucleus, I mean, is huge, and there's only a halo of cytoplasm, okay? But this is a lymphocyte. And what we're gonna make here is B cells, and T cells. And this is what we call a T helper cell. So you can write that down. Okay, this is called a T helper cell, which is a lymphocyte. So if you remember the blood section, when we talked about different lymphocytes, we had B cells and we had T helper cells. Okay, B cells and T helper cells. Okay. All right, so we finished. We're not done with the neutrophil, but we're going to come back to him, okay? So we've let the antigens out. They've gone to the lymphatic system. Then they've floated and drifted up into the lymph node, okay? All right, so now let's go to neutrophils first. Now, the neutrophil sent out some chemotaxis, which is chemical messengers that attract other white blood cells. It causes the capillary beds, okay? It causes the capillary beds to become sticky at the area of infection. They become sticky to other white blood cells, okay? So that's the chemotaxis, that's what the neutrophils do. So as the macro, or the monocytes, as they come by, they get stuck on the capillary beds that have swollen, okay, to bring more fluid. Then what happens? Those monocytes cross from the bloodstream over to the site of infection, all right? For the site of infection, which is called what? Diaphoresis, or immigration with an E, not an I, not like immigration like coming across the border, like E as an immigration, okay? All right, so immigration occurs, the monocyte becomes the macrophage, the tissue is swollen, the ground substance has become thin and watery, and that macrophage, which is not as motile as a neutrophil, but it can then come up through the tissue and get to where all these happy little bacteria are. Okay, this is a bacillus. And you guys taking micro right now? Yeah. Anyone? Yeah, there's a bacillus, a happy little bacillus right with the tail. Okay. All right. Okay, so then the macrophage is there. It goes through the same process. Remember, it's a macrophage, so it's a big eater. Okay, it brings in, through a process of endocytosis or phagocytosis, it brings in the bacterium as well. All right, lysosomes digest the bacterium just like before, okay? And then what we have is this phagosome with these star antigens in it. And that's it. Okay, now, this is where it changes. It doesn't just throw the antigens out into the system, okay? What we're going to create is what we call a T helper cell, right? And they call it a T helper cell because it's going to help your immune system, okay?
Bless you. Thank you.
Okay. All right, now, so what happens is, in the macrophage, after it's been stimulated by going through phagocytosis, chemical messengers will start the machinery of the nucleus to start working, all right? And what it's doing up here in the nucleus of the macrophage, in chromosome number 6, first of all, what's the job of the nucleus?
Control center.
Okay, so the control center does what? Someone else. What's the main job of the nucleus? It makes what?
A DNA.
It doesn't make DNA. It makes RNA in the process of making what? Protein synthesis, right?
Yeah.
Okay, so protein synthesis is the job of the nucleus, okay? So it does that by creating a copy of the DNA called messenger RNA. All right, now messenger RNA is a code. It's not anything yet, it's just a code, right? All right, it's the DNA code represented as messenger RNA. And then from messenger RNA, it goes through leaves the nucleus through the nuclear pores. It goes to the ribosomes, free and fixed. What? Okay, so where then it'll start linking amino acids to make the protein, right? Okay, now, what changes in here is the upper portion in a gene. So what we have is a gene, all right? And the upper portion of the gene will create different shapes, where the bottom portion of this gene will keep the protein stable. Okay, so I want you to understand this. So this gene codes messenger RNA that makes a protein where the top portion of that protein has a shape to it. Remember, what's the most important thing about proteins is the shape, right? The bottom portion stays the same, okay? And it keeps going through that. It shuffles through. It's going through this random process. And every one of these random messenger RNAs gets converted into a protein. That protein is then sent here, okay, into the phagosome, where then what happens is that protein will check to see if it attaches to the antigen. Remember, lock and key, okay, right? One has to fit the other. It's all about shape. Okay, so this antigen is a protein or peptide, right? Peptide antigen, okay? All right, so then this comes down, and a star antigen, okay, needs this shape. This is it right here, okay? Needs this shape to fit on there. What it's going to do is going to send this shape, this shape, this shape, and it's going to keep sending shapes down there until it finds one that fits. It's totally random. Okay, so it shuffles through chromosome number 6, changing its shape. Okay, changing its shape. Then what it does, it sends all these down there and keeps checking it and checking it and checking it and checking it. Eventually, it finds one that fits. Now remember, you have to take a step back and look big picture and remember what I'm telling you. Bacterium are multi-antigenic, okay? So it's not just one antigen that this is occurring to. This is lots, 28 I think is what the typical number is, I think is what they said. Okay, so we have a whole bunch of antigens, a whole bunch of messenger RNA. This doesn't do one at a time, okay? As we think of like, well, here's one, and here's another one, and here's another one. We're talking about putting out millions of them quickly, okay? So eventually you're gonna find a shape. And it only takes, you know, how long does it take your immune system to get fired up? You know, going through the whole process of actually really fighting infection, 14 days to get rid of something, right? 7 to 8 days to actually find stuff. So you have time for this. That's why it takes long. That's why it takes less time when you've had something to fight it already, because you already have the memory for this, okay? All right. All right, so anyway, so once it finds something, then the macrophage is gonna be a happy macrophage. All right, it's going to leave the site of infection, and it's going to enter the what?
The lymph vessel.
The lymph vessel, then go to the
Lymph node.
lymph node. In the lymph node, the macrophage, okay, is as on its process, it's going to take this antigen and this molecule and express it on the outside. When I say express, I just mean puts it on the outside of the macrophage, okay? It's going to go into the lymph node, and it's going to start finding lymphocytes. Okay, remember this is a T helper cell. Okay, T helper cell. Okay. Now, let me tell you that this molecule that's being generated in chromosome number 6, okay, is called MHC2, MHC class 2 molecule. This stands for major histocompatibility class 2. And when I say histo, what does histo mean? What does that mean? What is histology? Tissue.
Tissue.
Okay, so histology is the study of tissue. And then compatibility, all right? So what does that mean to us? Major histocompatibility class 2.
Tissue compatible.
Tissue compatibility, which means what? Which means MHC2 class 2 molecules are the reason why you can't take somebody's kidney and just slap it into somebody else, okay? Because your tissue will not accept it, right? So we have to cross-match tissue, and you have to be a match before you can get a kidney, right? Everyone knows that. You can't just give someone a kidney or a heart or all that stuff. Unless, of course, you do what? What do you have to do to the patient if you're just gonna give them any old kidney? You have to give them immune suppressant drugs. All right, and what do those drugs fight? They actually block the MHC2 molecule. Okay, which of course, if you do that, you're doing what to your immune system?
Killing
You're kicking its butt, and you don't have an immune system anymore, which is bad. Okay, because everywhere around you, on every surface around you, on everything you own is bacteria. All right, and our immune system is a wonderful tool that will, see, she's drinking a whole bunch of bacteria right now. Okay? All right, there's no way around it. Okay, so it's there all the time. But, so that's what that is. Okay, so now we take this macrophage has gone into the lymphatic node. We have expressed on the outside of the macrophage, we have a antigen and a molecule called an MHC2. Okay, major histocompatibility molecule, class 2. All right, now inside the lymph node there are these mature lymphocytes, okay, and they are called T cells, T helper cells. And where do those T helper cells mature? Starts with a T.
tissue. Oh, no.
The thymus. Very good. Okay, so they mature in the thymus, that's why they're called T cells, okay? They mature in the thymus.
Oh, and if it's in the bone marrow, then the B cells.
You got it. Okay, so the T cells are matured in the thymus. That maturation process actually starts creating this right here. This is called the TCR, T cell receptor. Now, that T cell receptor, every single cell has a different T cell receptor. So some T cell receptors for another T cell may have a T cell receptor that is like that. And some may look like that. And some may be that. Okay? It's completely random. So if these receptors that are on the outside of this T cell receptor, if the antigen never shows up that fits that, it never shows up, that T cell will not be activated. Okay, it just sits there, hangs out, waits patiently for a particular receptor. Okay, so this is where the presenting cell comes in. So this macrophage then will go to different T cells and start presenting two objects to each T cell. One is the what? No, that macrophage is presenting. So what is it presenting? It's presenting two things. It's presenting the, don't guess, it's right there. It's presenting the antigen and the other thing, right next to it.
T cell.
MHC2. Okay? It has to, it's presenting this to every single T cell it comes across to activate that T cell. Okay? T cells will not activate with just an antigen. Okay? They must have the MHC2 presenting molecule. Okay? So they must have this molecule. So we have the antigen and MHC2, this macrophage going to this one. Oh, nope, doesn't fit there. Doesn't fit there. Doesn't fit there. Nope. And it keeps going until it finds one. And then it says, yeehaw. Okay? Now, once it finds one, okay, the macrophage goes, all right. So now, what it does is once these two are stimulated, this thing on the inside of the T cell activates. It's called CD3. Activates and sends a signal down to the nuclear machinery, to a gene. Okay? CD3, now anytime it's CD, it just means cluster designation. When they were looking through an electron microscope, they saw a cluster of this tissue on the cell. So they just called it a cluster of cells. So they ended up just numbering them, so they call it cluster designation, in this case 3. Well, the MHC2 molecule attaches to CD4, all right? Right, so we have, so that you know, on the outside of the T helper cell, we have a TCR receptor, or TCR, which is T cell receptor, okay? And we have a CD4. That's the MHC2 receptor. I guess we can write that like that too. So this is, you can call this CD4 is also known as what? MHC2 receptor. Okay? Does that make sense? MHC2 receptor. Okay, now those four things together, not including CD3, those four things together, antigen, MHC2 molecule, CD4, and TCR all make up what's called the first signal or primary signal. Okay, that's the first signal. Once that has occurred, this macrophage will send out communication, and what do we call hormonal communication? Crin, C-R-I-N. Remember, guys? Okay, so remember merocrine, right? And then so this is an autocrine stimulation. Okay, I'm sorry, this is an autocrine, but this is an interleukin-1 is a interleukin is communication from one cell to the next. Okay, all right. So this cell will actually send out communication from one white cell to the next cell. Okay, and this is what we call and what are white blood cells called? Leukocytes. So this is interleukin-1. All right. So this stimulation goes to interleukin-1 goes to interleukin-1 receptor on the outside of this T cell. Does that make sense? Everyone okay with that so far? All right. Well, and then of course once you stimulate interleukin-1 receptor, a signal is sent down to the nuclear machinery of this cell. And then at the same time, right after this has occurred, we have another signal that comes down, and actually an appendage comes out of the macrophage and attaches to another receptor on the outside of the T cell, and that is called CD28. Of course, this is B7. So B7 and CD28 make up the second signal, or what we call co-stimulation. Okay? So that's co-stimulation or second signal. All right, now, the reason you go through all of that craziness right there is because we don't want what to occur. This is safety, because we don't want what to happen. We don't want our immune system doing what? Attacking itself or us, okay? So you have to go through this process to make sure that you don't kill yourself with your immune system, okay? And people that have degenerative problems or congenital problems with their immune system can go through stuff like this, which is terrible, okay? All right. So here we are. So we have first signal, which is MHC2, class 2 molecule, the antigen, which attaches to CD4 and TCR. That's the first signal, primary signal. That stimulates CD3, which stimulates the nuclear machinery. Interleukin-1, which stimulates interleukin-1 receptor, which stimulates the machinery. And B7, which stimulates CD28, which is, and you can even call this B7 receptor if you wish, okay? Makes this, right? Which then stimulates the nuclear machinery. Right now, you must have all three of those before the very first gene will start producing from the nucleus, okay? And that very first gene from the nucleus will send a signal which will send out a communication, all right? It's called interleukin-2, all right? Interleukin-2, which then comes back, and the second gene expresses a receptor on the surface of itself, interleukin-2 receptor. So then this interleukin-2 comes over and stimulates this interleukin-2 receptor, which then sends a signal back to the nuclear machinery, and this third gene is where the show starts. Okay? So, got it? So, once you've stimulated all three times with first signal and co-stimulation, okay, then interleukin-2 receptor, which is an autocrine stimulation, okay, stimulates itself, which then sends a signal back to the nuclear machinery, and this right here starts what we call mitosis. All right, mitosis. And what is mitosis? What? Making of cell. It's not wrong. Just making of the cell, so reproduction,
Yeah.
right? Okay. So it's making a clone of itself, right? So this guy is for mitosis. So, cellular reproduction, okay? All right, so now, what's the point of going through mitosis? Why? Why do we go at this third gene inside here? Why do we start mitosis? What are we trying to do?
More cells with the answer.
Okay, so we have to kill the infection. Now, infections, does it involve one bacillus? One guy cruising around?
No.
Millions and millions and billions of cells, depending on how bad the infection is, right? So one cell is not gonna get the job done, okay? Why do lymph nodes swell? Is that the site of the infection? No, not
Because they
necessarily.
bring it to it,
No.
so
Is that because they're full of interstitial fluid?
No. Well,
they bring the
yes,
infection
partly.
in there.
Is it more
Look.
cells?
Right here, clonal expansion, okay? So when we're making a clone of this, we're going through this thing called clonal expansion, and the cells are multiplying and multiplying, and we're making millions and millions of cells that are all identical to this. And why is that important? Because we're fighting
infection.
with
The T cell receptor,
the T cell receptor. Why is that important? Because it fits what?
the antigen.
The antigen for this infection. Does that make sense? That's the job. That's what we're trying to do. So we have to get these stimulated so they'll all release the same stuff. Okay, so now, once we go through mitosis, all right, this will start reproducing. Also, fourth and fifth genes will start releasing interleukin, right? Interleukin. Now, what's after 2? Interleukin, there's gotta be an interleukin 3, right? But they're not gonna make it that easy for you. Interleukin 3 goes off and messes with the bone marrow, all right, B cells. Interleukin 4, we're not gonna get into that. That takes more time than we have time left in the semester for. All right, so interleukin 4 and 5 come from the fourth and fifth gene, okay? Interleukin 4 and interleukin 5. All right, so we're going through clonal expansion. All of these cells are producing interleukin 4, interleukin 5. All of these cells are producing interleukin 2, which is autocrine stimulation, which can also stimulate the interleukin 2 receptors of the other cells. All right, so we have this big mess of stuff going on, which is causing mitosis to occur, which is causing clonal expansion, which is causing your lymph nodes to swell, along with the B cells. Okay, going through clonal expansion, all these cells are sending out interleukin 4 and interleukin 5. Okay, so interleukin 4 is what we call B cell growth factor. Okay, interleukin 4 is what we call B cell growth factor. Okay, B cell growth factor. Interleukin 5 is B cell differentiation factor. Interleukin 5, what is differentiation? Changing. Specializing, that's fine. Okay, so differentiation is where a cell will change. Okay, we talked about it before, where we actually have stem cells will change into the cell that they need to be. That's called differentiation. Okay, or germinative cells. That's the same thing as stem cell. Okay, all right. So, interleukin 4, interleukin 5. Interleukin 4 is B cell growth factor. Interleukin 5 is B cell differentiation factor. Now let's stop there for just a second and go back up there to Mr. Neutrophil. Okay, Mr. Neutrophil did what he do with his antigens. Just right, he's lazy. All he did was release those antigens off into the tissue. All right, and that tissue then picked up the antigens, took it through the lymphatic system, through to the lymph nodes. Okay, where in the lymph nodes it comes across a B cell. The B cell is also a lymphocyte, right? Okay. Now, where B cells mature? Bone marrow. B cell for bone marrow, right? Okay. Now, when they're maturing, they're kind of doing the same thing that the T cells did. They are creating receptors on the outside of the B cells that are completely random in nature. Okay. So B cells are going through B cell receptors that are completely random in nature. So as they're maturing, it's just making more and more and more and more with different receptors on them. And eventually, right, this antigen is gonna wash across a receptor that it fits into, okay? And once that occurs, that B cell is stimulated. There's a cluster designation called CD79 on the inside that's stimulated. Sends a signal down to the nuclear machinery, which then expresses a receptor on the outside of the cell called interleukin-4 receptor. Okay? Now these are called T helper cells here, right? Right here's where they help. So all that interleukin-4 that we're releasing from these cells then comes over here, attaches to these receptors here, which causes the nuclear machinery to go crazy, all right, and start going through mitosis. And this is what we call mitotic storm, because there's receptors all the way around this cell. Interleukin-4 is washing over these cells, and all of a sudden this cell is going crazy with separating. Okay, so that's what stimulates the mitotic process to go on. Once you go through the mitosis, another receptor is expressed on the outside, interleukin-5 receptor, and interleukin-5 was called B cell differentiation factor, okay? And that causes the B cells to differentiate into a plasma cell. Okay, causes the B cell to differentiate into a plasma cell. All right, and plasma cells end up with a very large, what am I trying to draw here? What is that right there? That's a Golgi apparatus, our Golgi bodies. And what does Golgi do?
They
Transport FedEx.
FedEx, right? Packaging and shipping, okay?
That's-
Packaging and shipping. So the Golgi apparatus ships out these things called antibodies. And everybody's heard of an antibody, right? Okay. Antibodies are part of your immune system. They are Y-shaped like this, okay? And each of the ends of the Y shape are very specific. They have the shape of
the antigen,
which antigen?
the MH, no.
Nope, that's a class 2 molecule. Which antigen? These
The
antibodies.
blood.
What?
Of the blood.
No, no. Which antigen is this the shape of? This is the whole reason
The
for doing all
infection.
this. The infection. So it's the shape of what? The star.
Yeah.
The star antigen, right? That's the reason for doing all this. That's why we went through all this process with the very specific molecules, very specific process. This antigen attaches, this goes crazy. So when you make these antibodies here after this process, the ends of these antibodies fit that star antigen. Again, remembering, please, that it's not one antigen this occurs for, it's multi-antigenic, okay? All right, so now the antibodies are what cause complement to show up. And I took complement out of our lectures for time's sake. Complement would take us another class period to do, okay? So, but what happens is we actually put like a flag here, which starts attracting more proteins. We end up with this thing that kind of comes down towards the bacterium and then punches a hole in the bacterial cell. Okay, so which, of course, if you punch a hole in some living organism's body, it does what? It dies, right? The stuff expels. It's called lysis in cells, okay? So complement, what does complement do? It's the name. It complements
The infection.
the immune system, okay? So normally you guys have to remember, what is the immune system? We have natural killer cells that are running around. We have phagocytes, macrophages running around, neutrophils, dendritic cells. All of these are phagocytic cells that are in our system all the time, and they don't necessarily need to go through this to start affecting something that is not self, all right? But if something's there and has a foothold, like an infection, we have to go through this process to make sure we get rid of it, okay? But if it's not self, we don't necessarily need complement if we can get rid of it with the other cells. But this complements the the immune system, okay? This right here is complement, okay? And then the next thing it does is, these guys, I show you a bacillus because it has a tail, which means it does what? It moves, okay? So if it moves better, faster, can be faster than neutrophils, which are fast, and macrophages. So those guys gotta catch it, okay? So if I take an antibody and hold it down, tie it up, and then I waggle the tail, right? To entice a macrophage to come in, attaches to the tail of the antibody. So once it's held down, he grabs a hold of the rope of it and then starts to engulf it. Okay? So then that is where this will go through phagocytosis through the immune system. And this is called, when you tie it down and hold it, it's called opsonization. Okay? It's called opsonization. Okay?
Complement occur
Or opsonization. Okay? All right. Okay, that's it. That's all we're gonna cover.